Welcome to our continuing journey into the amazing world of human anatomy and physiology. Today, we dive deep into the very essence of life, the cell. Specifically, we will explore cell physiology, which is how our cells work, and then see how these individual units group together to form tissues. Every function in our body, from a simple thought to a powerful muscle contraction, begins at the cellular level. Understanding these microscopic powerhouses is fundamental to understanding ourselves. So let's begin by meeting the guardian at the gate of every cell, the structure that defines its boundaries and controls everything that goes in and out, the plasma membrane. It is a dynamic fluid barrier essential for life itself. The plasma membrane is a remarkable structure, often described as a phospholipid bilayer. Imagine a sandwich with two layers of bread facing each other. This is similar to how the phospholipids are arranged. Each phospholipid molecule has a head and a tail. The head is hydrophilic, meaning it is attracted to water. Since the environment inside and outside the cell is watery, these heads happily face outwards and inwards, towards the fluid. The tails, however, are hydrophobic, meaning they are repelled by water. They tuck themselves away from the water, facing each other in the center of the membrane, creating an oily, water-fearing core. This clever design creates a stable yet flexible boundary for the cell. This structure makes the plasma membrane selectively permeable. Think of it as a bouncer at a club. It doesn't just let anyone or anything in, it is very selective. It allows some substances to pass through easily while blocking others completely. Small, uncharged molecules like oxygen and carbon dioxide can slip through the oily core without any trouble. However, larger molecules like glucose or charged particles like ions, sodium, potassium, cannot get through on their own. This selectivity is crucial. It allows the cell to maintain a stable internal environment, a state known as homeostasis, which is very different from the environment outside the cell. The concepts of hydrophilic and hydrophobic are central to this function. As we mentioned, hydrophilic means water-loving, and these parts of the membrane interact with the water-based fluids inside and outside the cell. Hydrophobic means water-fearing, and these parts avoid water, forming the membrane's inner barrier. This dual nature is what makes the bilayer possible. The fluid inside the cell is called intracellular fluid, and it's rich in potassium and proteins. The fluid outside the cell is the extracellular fluid, which includes the fluid between cells, interstitial fluid, and blood plasma. Extracellular fluid is rich in sodium and chloride. This difference in composition creates an electrical and chemical gradient across the membrane, which is vital for many cellular functions, including nerve impulses and muscle contraction. Now that we understand the membrane structure, let's explore how substances actually move across it. The simplest way is through diffusion. Diffusion is the movement of particles from an area where they are in high concentration to an area where they are in lower concentration. Imagine opening a bottle of perfume in a room. At first, the scent is strong near the bottle, but over time, the perfume molecules spread out until they are evenly distributed throughout the room. This happens because molecules are in constant random motion. They naturally move down their concentration gradient from crowded to less crowded until equilibrium is reached. No energy is required from the cell for this to happen. Osmosis is a special type of diffusion that specifically involves the movement of water across a selectively permeable membrane. Like other substances, water also moves down its concentration gradient. It moves from an area with a higher concentration of water, which means a lower concentration of solutes like salt or sugar, to an area with a lower concentration of water or a higher concentration of solutes. Think of it this way: water moves to dilute the more concentrated solution. This movement creates a force called osmotic pressure, which is the tendency of water to move into a solution. This is balanced by hydrostatic pressure, the pressure of the fluid itself pushing against the membrane, like water in a balloon pushing outwards. The effects of osmosis are dramatically seen when we place a cell in different solutions. In an isotonic solution, the concentration of solutes outside the cell is the same as inside. Water moves in and out at an equal rate, and the cell maintains its normal shape. In a hypotonic solution, the solute concentration outside is lower than inside. Water rushes into the cell, causing it to swell and potentially burst, a process called lysis. Conversely, in a hypertonic solution, the solute concentration outside is higher than inside. Water moves out of the cell, causing it to shrivel or crenate. This is why giving a patient pure water intravenously is dangerous. It would cause their red blood cells to burst. Movement across the membrane can be categorized into a few types. Simple diffusion is when small, lipid-soluble molecules pass directly through the phospholipid bilayer, as we discussed with oxygen. However, many essential substances like glucose and ions are not lipid-soluble. They need help. This is where facilitated diffusion comes in. It still involves movement down a concentration gradient and requires no energy, but it uses protein channels or carriers embedded in the membrane to help the substances across. Finally, there is active transport. This process moves substances against their concentration gradient, from a low concentration to a high one. This is like pushing a ball uphill. It requires energy, which the cell provides in the form of a molecule called ATP. Active transport is a critical process that allows cells to accumulate necessary substances or expel waste products against the natural flow of diffusion. The most famous example of an active transport system is the sodium-potassium pump. This protein pump is found in the membrane of virtually all animal cells. It actively pumps 3 sodium ions out of the cell for every 2 potassium ions it pumps in. This process requires energy, which is supplied by breaking down a molecule of adenosine triphosphate. By constantly working, this pump maintains the high concentration of potassium inside the cell and the high concentration of sodium outside, which is essential for nerve impulses and muscle function. Sometimes the cell needs to transport very large particles or large quantities of small molecules that cannot pass through the membrane or a protein channel. For these situations, the cell uses a process involving vesicles, which are small, membrane-bound sacs. This process is generally called vesicular transport. Endocytosis is the process by which the cell takes material into the cell by engulfing it. The plasma membrane folds inwards, forming a pocket around the substance, which then pinches off to become a vesicle inside the cell. This method allows the cell to eat and drink from its environment. There are a few types of endocytosis. Phagocytosis, which means cell eating, is when the cell engulfs large, solid particles like bacteria or cellular debris. This is a key function of immune cells like macrophages. Pinocytosis, or cell drinking, is when the cell gulps a drop of the extracellular fluid containing dissolved solutes. It's a routine way for the cell to sample its environment. Exocytosis is the reverse process. It's how the cell expels substances. A vesicle containing the substance, such as hormones, neurotransmitters, or waste, moves to the plasma membrane, fuses with it, and then ruptures, releasing its contents outside the cell. These transport mechanisms are fundamental to cellular life. They allow for the selective intake of nutrients, the removal of waste, communication between cells, and the maintenance of the precise internal environment required for all other cellular processes to function correctly. From the simple diffusion of gases to the complex, energy-driven pumping of ions and the large-scale engulfing of particles, the plasma membrane and its associated proteins work tirelessly to manage the constant traffic that sustains the cell. It's a bustling city gate with goods and messages constantly flowing in and out, all orchestrated with incredible precision. Inside the cell, a vast number of chemical reactions are constantly occurring, collectively known as metabolism. These reactions would happen far too slowly on their own to sustain life. To speed them up, the body uses biological catalysts called enzymes. Enzymes are magnificent protein molecules that increase the rate of a chemical reaction without being used up in the process. Think of an enzyme as a specialized tool on an assembly line. It helps to put two pieces together or take them apart much faster and more efficiently than you could with your bare hands. And once the job is done, the tool is ready for the next task. Each enzyme is highly specific. It typically binds to only one type of molecule, called its substrate. This specificity is due to the enzyme's unique three-dimensional shape, which includes a region called the active site. The active site is shaped to perfectly fit its substrate, much like a lock and key. When the substrate binds to the active site, it forms an enzyme-substrate complex. This interaction causes a slight change in the enzyme's shape, which puts stress on the substrate's chemical bonds, making it easier for the reaction to occur. For example, the enzyme amylase, found in our saliva, has an active site that perfectly fits starch, a carbohydrate, helping to break it down into smaller sugars. The rate at which enzymes work is influenced by several factors, including temperature and pH. Most enzymes in the human body function optimally at normal body temperature, around 37 degrees Celsius or 98.6 degrees Fahrenheit. If the temperature gets too high, the enzyme structure begins to break down. The weak bonds holding the protein in its specific three-dimensional shape are disrupted, and the enzyme loses its shape. This process is called denaturation. A denatured enzyme's active site is altered, so it can no longer bind to its substrate. This is irreversible, like cooking an egg. You can't uncook it. This is why a very high fever can be life-threatening. It denatures critical enzymes. Many enzymes also require non-protein helpers called cofactors or coenzymes to function properly. Many vitamins act as coenzymes. They bind to the enzyme and assist in the reaction, often by helping to transfer atoms from one molecule to another. This is why a balanced diet rich in vitamins is so essential for our health. Without them, many of our vital metabolic reactions would grind to a halt. In summary, enzymes are the indispensable workforce of our cells, facilitating the thousands of chemical reactions that keep us alive, and their proper function depends on maintaining a stable internal environment. The instructions for building and operating the entire body, including making all those vital enzymes, are stored in a remarkable molecule called deoxyribonucleic acid, or DNA. DNA is the blueprint of life. It is an organic compound, which simply means it is a large carbon-based molecule. It is a double helix, resembling a twisted ladder, and it resides safely within the nucleus of our cells. The rungs of this ladder are made of 4 chemical bases: adenine, guanine, cytosine, and thymine. The specific sequence of these bases forms a code that dictates everything from your eye color to how your cells produce energy. Because DNA is so precious, it never leaves the nucleus. To get its instructions out to the cell's protein-building machinery, the ribosomes in the cytoplasm, it relies on a messenger molecule called ribonucleic acid, or RNA. RNA is similar to DNA but has a few key differences. It's a single strand, not a double helix. It uses the sugar ribose instead of deoxyribose, and it uses the base uracil instead of thymine. The process of creating an RNA copy of a DNA gene is called transcription. This messenger RNA molecule then travels out of the nucleus to a ribosome. At the ribosome, the process of translation occurs. The ribosome reads the sequence of bases on the mRNA in groups of 3 called codons. Each codon corresponds to a specific amino acid, the building blocks of proteins. Another type of RNA, called transfer RNA, is responsible for fetching the correct amino acid and bringing it to the ribosome. As the ribosome moves along the mRNA, a chain of amino acids is assembled in the precise order dictated by the original DNA code. This chain then folds into a specific 3-dimensional shape to become a functional protein, such as an enzyme, a hormone, or a structural component of the cell. This intricate flow of information from DNA to RNA to protein is known as the central dogma of molecular biology. It is the fundamental process by which our genetic blueprint is translated into the functional components that make up our bodies and carry out the processes of life. Organic compounds, particularly proteins, made of amino acids, carbohydrates, lipids, and nucleic acids, form the structural and functional basis of all living things. Understanding how DNA and RNA orchestrate the synthesis of these molecules is key to understanding health and disease. Nerve cells, or neurons, are the communication specialists of the body. They transmit information as electrical signals, allowing for rapid communication between different parts of the body. To be ready to send a signal, a neuron must first be in a resting state. In this state, the neuron's plasma membrane is polarized, meaning there is an electrical voltage difference across it. The inside of the membrane is negatively charged relative to the outside. This voltage is called the resting membrane potential, and it is typically about -70 millivolts. This negative charge is like a coiled spring, storing potential energy that can be released to generate a nerve impulse. Several factors contribute to establishing and maintaining this resting membrane potential. First, the intracellular fluid has a higher concentration of negatively charged proteins and phosphate ions compared to the extracellular fluid, contributing to the negative interior. Second, and most importantly, is the differential permeability of the membrane to potassium and sodium ions. The membrane has many more potassium leak channels than sodium leak channels. These channels are always open, allowing ions to diffuse down their concentration gradients. Since there are many potassium leak channels, potassium ions, which are highly concentrated inside the cell, readily leak out, taking their positive charge with them. The outward leak of positive potassium ions is the primary reason the inside of the cell membrane becomes negative. While some positive sodium ions do leak into the cell through their own few leak channels, the outward flow of potassium is much greater. This continuous leakage would eventually disrupt the concentration gradients, but the cell has a mechanism to counteract it: the sodium-potassium pump. As we learned earlier, this active transport pump uses ATP to constantly pump 3 sodium ions out for every 2 potassium ions it brings in. This action is electrogenic, meaning it contributes a small amount to the negative charge inside, but its main role is to maintain the concentration gradients that the leak channels rely on. So, to summarize, the resting membrane potential is a product of 3 main factors: the presence of negatively charged proteins inside the cell, the much greater permeability of the membrane to potassium than to sodium due to potassium leak channels, and the continuous action of the sodium-potassium pump, which maintains the concentration gradients for these ions. This carefully maintained state of readiness is crucial. It ensures that the neuron is primed and ready to fire an electrical signal, known as an action potential, as soon as it receives the right stimulus. The part of the neuron that typically receives these initial signals is the dendrites. When a neuron is stimulated, its resting state can be disturbed. Incoming signals from other neurons can cause small, localized changes in the membrane potential. An excitatory postsynaptic potential, or EPSP, makes the inside of the membrane slightly less negative. This is called depolarization, and brings it closer to the threshold for firing. An inhibitory postsynaptic potential, or IPSP, makes it more negative or hyperpolarized, moving it further from the threshold. If enough EPSPs occur to depolarize the membrane at the axon hillock, the region where the axon joins the cell body, to a threshold level, usually around -55 millivolts, an action potential is triggered. This is an all or none event. It either happens completely or not at all. An action potential is a rapid, temporary reversal of the membrane potential that propagates along the axon of the neuron. It involves the opening and closing of voltage-gated ion channels. The first step is depolarization. Once the threshold is reached, voltage-gated sodium channels burst open. Since sodium is highly concentrated outside the cell, sodium ions rush into the cell, and the massive influx of positive charge causes the membrane potential to shoot up from -70 millivolts to about +30 millivolts. The inside of the membrane briefly becomes positive relative to the outside. This rapid change is the nerve impulse itself. This depolarized state is very brief. The sodium channels inactivate and voltage-gated potassium channels open. This marks the beginning of repolarization. Now, with the potassium channels open, potassium ions, which are highly concentrated inside the cell, rush out, carrying their positive charge with them. This outward flow of positive ions causes the membrane potential to fall back down, returning towards the resting potential. In fact, these potassium channels are a bit slow to close, so for a short period, too much potassium leaves the cell, causing the membrane potential to dip slightly below the resting potential. This brief dip is called hyperpolarization. During and immediately after an action potential, the neuron enters a refractory period. The absolute refractory period occurs during depolarization and repolarization, when the sodium channels are either open or inactivated. During this time, the neuron cannot generate another action potential, no matter how strong the stimulus. This ensures that each action potential is a separate, all or none event. This is followed by the relative refractory period during hyperpolarization. Here, a new action potential can be generated, but it requires a much stronger stimulus than usual. This entire sequence of events propagates down the axon like a wave. In myelinated axons, this process is much faster. The myelin sheath, which is a fatty insulating layer, prevents ion leakage. The action potential jumps from one gap in the sheath, a node of Ranvier, to the next. This process is called saltatory conduction, and it greatly increases the speed of nerve impulse transmission. Cells are the basic units of life, but they rarely work alone. In complex organisms like humans, cells with similar structures and functions group together to form tissues. Tissues represent the next level of organization in the body, and they are the building blocks of all our organs. The study of tissues is called histology. There are 4 major types of tissue in the human body, each with a distinct role: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Understanding these 4 types is fundamental to understanding how our organs are constructed and how they function as part of a larger system. Epithelial tissue, or epithelium, covers body surfaces, lines body cavities and hollow organs, and forms glands. Its cells are packed tightly together in continuous sheets with very little extracellular material between them. This close-knit structure makes it an excellent protective barrier. Epithelia always have one free surface, called the apical surface, which is exposed to the body's exterior or the cavity of an internal organ. The lower surface, called the basal surface, rests on a basement membrane, a thin layer of material that anchors the epithelium to the underlying connective tissue. Epithelia are also avascular, meaning they have no blood supply of their own. They rely on diffusion from the capillaries in the underlying connective tissue for nutrients and oxygen. There are many types of epithelia classified by cell shape and layer arrangement. Simple epithelia have a single layer of cells ideal for absorption, secretion, and filtration. Simple squamous epithelium, a single layer of flat cells, is found where filtration or rapid exchange of substances is critical, such as in the air sacs of the lungs and the lining of blood vessels. Simple cuboidal epithelium, with cube-shaped cells, is common in glands and their ducts, like the salivary glands and the walls of kidney tubules. Simple columnar epithelium, made of tall column-like cells, lines the entire digestive tract, where it is involved in secretion and absorption. The apical surfaces of these cells often have microvilli to increase surface area. Stratified epithelia consist of 2 or more cell layers, making them durable and ideal for protection. Stratified squamous epithelium is the most common type, found in areas subject to abrasion, such as the outer layer of the skin and the lining of the mouth and esophagus. Transitional epithelium is a highly modified stratified epithelium found only in the urinary system organs, like the bladder. Its cells can slide past one another and change shape, allowing the organ to stretch as it fills with urine. 2 other special types are pseudostratified columnar epithelium, which appears stratified but is actually a single layer of cells of different heights, and glandular epithelium, which forms the glands of the body. While epithelial tissue provides covers and linings, connective tissue is the most abundant and widely distributed tissue type in the body. As its name implies, its primary roles are to connect, support, bind, and protect body organs. Unlike epithelial tissue, connective tissues are typically characterized by having a large amount of non-living extracellular matrix that separates the living cells of the tissue. This matrix, produced by the connective tissue cells, is what allows connective tissue to bear weight, withstand tension, and endure abuses that no other tissue could. The matrix consists of a ground substance, which is a watery, gel-like material, and fibers, collagen, elastic, or reticular. Connective tissues vary widely in their structure and function. They include bone, cartilage, dense connective tissue forming tendons and ligaments, loose connective tissue like areolar and adipose tissue, and even blood. Areolar tissue is a soft, pliable, cobweb-like tissue that cushions and protects the body organs it wraps. Adipose tissue is commonly known as fat. It forms the subcutaneous tissue beneath the skin, where it insulates the body and protects it from bumps and extremes of heat and cold. Cartilage is less hard and more flexible than bone. It is found in structures like the larynx, the tip of the nose, and the joints. A major characteristic of cartilage is that it is avascular, which is why it heals very slowly when injured. The cells found within connective tissue are diverse and include fibroblasts, which build the fibers, chondrocytes in cartilage, osteocytes in bone, and various cells involved in defense, like macrophages and white blood cells. Blood is considered a connective tissue because it consists of blood cells, red and white, surrounded by a non-living fluid matrix called plasma. The fibers of blood are soluble protein molecules that become visible only during clotting. It serves as the body's main transport system, carrying nutrients, wastes, hormones, and respiratory gases throughout the body. Finally, glands, which produce and secrete specific products, develop from epithelial tissue but are often embedded within connective tissue. There are 2 main types, exocrine and endocrine glands. Exocrine glands retain their ducts, and their secretions like sweat, oil, saliva, and digestive enzymes are emptied through these ducts to the epithelial surface. Endocrine glands are ductless. Their secretions, called hormones, diffuse directly into the bloodstream to travel to target organs. Exocrine glands can be further classified by how they secrete their products. Merocrine glands, like most sweat glands, secrete via exocytosis without damaging the cell. Apocrine glands accumulate products at the cell's apex, which then pinches off. Holocrine glands, like the oil glands of the skin, accumulate products until the entire cell ruptures. Understanding these cellular and tissue-level structures is the key to appreciating the complex and integrated machine that is the human body.